Hi, welcome back. This is Anatomy and Physiology 1, Lecture 2, The Cell Structure. This is part 2 of 2. Disclaimer. Welcome to Anatomy and Physiology 1, Lecture 2. I have put together a series of A&P1 presentations that will be posted at later dates. I hope this will help A&P1 students. Please take your time while reviewing this lecture. You may use your textbook to cross-reference. To have a clear understanding of this subject, I suggest that you review this presentation before and after you have done your reading. Do not use this presentation as your only source of learning. Listen to your teacher/instructor, read your textbook, and take notes. This slide is for entertainment and educational reference only. I am not a teacher. Please see your teacher/instructor for any questions and answers that have not been addressed here. The cell interior. After reviewing this section of the slides, you should be able to list the main organelles of a cell, describe their structure, and explain their functions. You should also be able to describe the cytoskeleton and its functions, as well as give examples of cell inclusions and explain how inclusions differ from organelles. The cytoskeleton. The cytoskeleton is a network of protein filaments and cylinders. The cytoskeleton determines cell shape, supports the structure, organizes cell contents, directs movement of materials within the cell, and contributes to movements of the cell as a whole. It is composed of microfilaments, intermediate fibers, and microtubules. Now let's touch a little bit on the components of the cytoskeleton. Okay, microfilaments. Microfilaments are made up of a protein called actin. Microfilaments help generate forces used in cellular contraction and basic cell movement. They enable the dividing cell to pinch off into two cells. Okay, actin works together with another protein called myosin to produce muscle movement. It also assists in cell division as well as cytoplasmic streaming. And it also assists or helps with endocytosis and exocytosis. So basically, microfilaments helps the cell with shape, support, stability, and movement. Intermediate fibers. Okay, so the intermediate fiber, their role basically is to provide mechanical strength to cells and tissues. It prevents the fracture of epithelial cell sheets under tension. So intermediate fibers are found strongly in hair. Examples would be in hair and nails. Then you have the microtubules. What the microtubules do is they regulate cell growth and movement. They provide the cytoskeleton that gives the shape to many cells. Okay, they are a major component in cilia and flagella. Remember the flagella I was telling you that's found only in humans, which would be the sperm, the tail at the end? Well, that's where the microtubules would be found, okay? Also, they participate in the formation of the spindle during cell division. Hi, so here is a picture of a cell. And this is the cytoskeleton of the cell. So remember in the last part of the lecture, I was telling you about macrovilli, and I tried to draw a picture. Well, don't laugh at my drawing, but here's a nice picture of the macrovilli, and what it does is it gives the cell more surface area, okay? Now, if you look at the microvilli, here's some microfilaments. See how this line that goes up, around, and down? That is the macrofilaments, okay? And these fine hairs, this is the intermediate filaments right here. And they go all over in this picture. They're all over. They're found all over. Then you have the microtubules. In this picture, this is the process of them assembling. And if you go on to the other side, here's the microtubules going under disassembly. Okay? Disassembly right here. Also, here is another picture. Here's another picture. These green, these are also intermediate filaments as well. So study the pictures. A lot of these things we'll talk about in future future slides. Hi, welcome back. This is just a quick review of the cytoskeleton. So the microfilaments are usually 6 nanometers thick. They're made of actin protein. Remember that when actin and the myosin together combine, they help produce movement of the muscle movement. They also form a terminal web. The terminal web is right here as well. And this is the microfilaments right here. This here. Okay. Remember on top here, that's the microvilli and the microfilaments. Okay, then you have the intermediate filaments. They're usually 8 to 10 nanometers thick. They're within the skin cells. They're made of a protein, keratin. Remember, the hair is made of keratin. That's what makes it strong. So the hair is the keratin. And these here, this is it, right? Intermediate filaments right here, right here. You have some more here. They give the cell shape and resist stress, make it strong. Then you have the microtubules, which is 25 nanometers thick. They consist of protofilaments made of a protein called tubulin. Now, tubulin is found only in eukaryotic cells, okay? They radiate from the centrome. This is the little centromes here. They can come and go. What else? They maintain the shape, hold the organelle, act as a railroad track for walking motor proteins. They make the axiomes of the cella and flagella, and they form the mitotic spindle. You know, sometimes I can't pronounce some of these things, but remember the flagella, the tail, the cilia, the little hair-like projections. The microtubules help form that stuff. Okay. Organelles. So you know how the human body has like the heart, the liver, the lungs, and so forth? Those are our organs. Well, in the cell's organs are called organelles. So the internal structures of a cell, and they carry out specialized metabolic tasks. There are 2 types, membranous organelles, meaning they have a membrane that surrounds them, and non-membranous, where they don't have a membrane that surrounds it. So the nucleus, mitochondria, lysosomes, peroxiomes, endoplasmic reticulum, and the Golgi complex, they have a membrane. The ones that don't have a membrane would be ribosomes, centrosomes, centrioles, and basal bodies. Hi, so we're here at the cytoplasmic organelle. So we have the plasma membrane, which goes all the way around right here. You have the systole, which is the intercellular fluid, which is the fluid within the cell. Then you have the exocytosis, which is, remember, when it wants to rid of waste or whatever. This is it. That's the little dimple that it forms after, the stuff has been expelled. It's right here. Here's the endocytotic transport vesicle, remember, when it wants to bring things in. So what it does is it sits here, this edge and this edge, they kind of form together like a little circle, see? And then it breaks off, and then it moves within the cell and brings whatever particles or solutes that it needs to be within the cell. Here you have the mitochondria, you have the free ribosomes, you have the smooth and rough endoplasmic reticulum. Smooth endoplasmic reticulum is called that because it doesn't have the ribosomes on there. And the rough endoplasmic reticulum is called rough because it has the ribosomes attached to it. Then you have, here's the nucleus, right here. This is the chromatin. It's kind of like a fluid within the nucleus. And then you have the envelope of the nucleus, and you have the nucleus itself, and then you have the nucleus pore, the nuclear pore right here. What else can I show you? I think I went over everything that was on here. This is the centrioles, and then the centromes as a combine. All of this here, these blue. Okay. So here we have the nucleus. The nucleus is supposed to be, they say it's the largest organelle. It's 5 microns. I can't even in diameter, but I can't even describe what 5 microns is. I would have to say it's invisible to the naked eye. That's what I would have to say. Most cells have one nucleus. There are a few that have no nucleus, anuclear, or some that have multi. Okay? You have nucleoplasm. That is the material in the nucleus. So you have the chromatin, which is a thread-like. It's composed of the DNA and protein. And then you have the nuclei, which is masses where ribosomes are produced. So here's something that you'll need to know. Ribosomes are produced in the nucleoplasm material in the nucleus, but it's here, masses where ribosomes are produced. So memorize this stuff. It kind of goes to the next things, okay? So here's a great picture of the nucleus right here. And then here's the nucleoplasm. That's the liquid and stuff inside the nucleus. And then you have the nuclear envelope, which is right here, that goes all the way around. And then you have the nuclear pores in this picture right here. That's really good picture. Oh, surface of the nucleus. And this is the interior of the nucleus. Endoplasmic reticulum. Endoplasmic reticulum is a system of channels called the cisternae enclosed by the membrane. This is the smooth endoplasmic reticulum. Then you have the rough endoplasmic reticulum. Remember I said the smooth endoplasmic reticulum does not have the ribosomes on them. However, the rough endoplasmic reticulum, they have the ribosomes. They're covered with ribosomes on them. Okay, they are attached. The rough endoplasmic reticulum is attached to the nuclear envelope. If you look at a picture, you see that the nuclear envelope, like the rough endoplasmic reticulum, it kind of swivels right off of it. The rough endoplasmic reticulum, they produce phospholipids and proteins of the plasma membrane, and they synthesize proteins that are packed in other organelles and secreted from the cell. Endoplasmic reticulum. So let's talk about the smooth endoplasmic reticulum. Well, they lack ribosomes, like I said before. The cisternae is more tubular and branching. The cisternae is taught to be continuous from the rough endoplasmic reticulum. So so here is the nucleus. We're going to draw a nice, I thought I was doing good. So this is the nucleus, okay? And from the nucleus, remember I told you that the endoplasmic reticulum, it kind of comes off the envelope of the nucleus envelope? Well, from there, it goes to the rough endoplasmic reticulum. And the rough endoplasmic reticulum, they have the ribosomes on there. The red little, the ribosomes on there. I'm making them red. Okay? And this follows it. It goes they're all over this thing. All over the endoplasmic reticulum wall. And then from the endoplasmic reticulum wall, from the rough side, then it becomes smooth, meaning they become smooth, and meaning they have no ribosomes on them, okay? So they also synthesize steroids and other lipids. They detoxify alcohol and drugs, and they store calcium. So the rough and smooth endoplasmic reticulum are functionally different parts, but they're part of the same network. Okay? So here is a picture of the endoplasmic reticulum. Here is the rough endoplasmic reticulum. This has all the ribosomes on there. See, it's labeled the ribosomes. And then here's the cisternae. This is the system that branches out. This is a system that it follows a pattern. And then from the rough enters the smooth, how it just becomes smooth endoplasmic reticulum. Ribosomes. Ribosomes are small granules of protein and RNA. RNA is ribonucleic acid. It is a nucleic acid present in all living cells that has structural similarities to DNA. We'll talk about that in a little bit more detail later on. Okay? Ribosomes are found in nuclei and in the cytosol and on the outer surfaces of the rough endoplasmic reticulum, and they're found on the nuclear envelope. They, quote unquote, they read coded generic messages and assemble amino acids into protein specified by the code. This is where things get a little complicated, but you'll see. We'll put it all together later. Let's discuss what synthesis is. Let me give you the definition of synthesis. The word is used so much, and sometimes I don't even think people really understand what it is. So let's just go over it real quick. It's basically the production of chemical compounds by reaction from simpler materials. That's it, okay? So, Golgi complex. The Golgi complex is a system of cisternae that synthesizes carbohydrates and puts the finishing touches on protein synthesis. The Golgi complex, what it does is it receives the newly synthesized proteins from the rough endoplasmic reticulum. All right, it sorts the protein and splices some. It adds carbohydrate. It packages them into a membrane. It makes them for what it's needed for. It kind of adds the finishing touches, like it was said. Some vesicles turn into lysosomes. Some migrate to plasma membrane and fuses to it. Some become secretory vesicles that store protein and production for later release. So here's the Golgi complex. Here's the Golgi vesicles. It will make whatever it has to make. Once it's it receives whatever it needs to receive from whatever protein or it receives from the rough endoplasmic reticulum, and then it makes it what it needs to. And these are the vesicles. After it's made, it's on the Golgi vesicle, and then it goes wherever it needs to go and attaches to wherever, like whether it's the membrane or other organelles within the cell or outside, or it stores things. It just depends on what its need for. Lysosomes. They are enzymes. They are enzymes that are bound by a membrane. They're usually round, but they can vary in shape. Their functions, they are intracellular hydrolytic digestion of proteins, nucleic acid, complex carbohydrates, phospholipids, and other substances. Autophagy, which is the digestion of cells' surplus organelles, and autolysis, cell suicide, digestion of a surplus cell by itself. These are the functions of the lysosomes. This is what they do. Peroxisomes resemble lysosomes, but they contain different enzymes and are produced by the smooth endoplasmic reticulum. Their functions is to use molecular oxygen to oxidize organic molecules. Reactions produce hydrogen peroxide, break down excess peroxide, neutralize free radicals, detoxify alcohol and other drugs, and a variety of blood-borne toxins, and they break down fatty acids into acetyl groups for mitochondrial use for them to make the ATP synthesis. They're in all the cells, but they are plentiful, they're abundant in the liver and kidney cells. Here's another picture. Here's a picture of the lysosomes and the peroxisomes. You can study it, look at it, so you have a clearer understanding. Here is a diagram of the proteasomes. Proteasomes, they are hollow, cylindrical organelles. They get rid of extra protein. It contains enzymes that break down the specific protein or whatever needs to be broken. And they target proteins, or they make them into like short peptides and amino acids. So you have mitochondria. Mitochondria are organelles that specialize for the synthesis of ATP ATP is adrenotriphosphate. ATP, what it is an energy carrying molecule found in the cells of all living things. ATP, it captures chemical energy obtained from the breakdown of food. Okay? The mitochondria is continuously changing shape. It goes from spheroidal to thread-like. It's surrounded by a double membrane. The inner membrane has folds called cristae, which would be, you see these things here, but here, this is the cristae. These are all cristae here. And within the cristae, the spaces of the cristae is called the matrix, okay? The matrix, it contains ribosomes, enzymes used for ATP synthesis, a small circular DNA molecule. Mitochondrial DNA, okay? The mitochondria is the powerhouse of the cell. Energy is extracted from organic molecules and transferred into ATP. Centrioles. They are short cylinder-like assembly of microtubules arranged in 9 groups of 3 microtubules each. When you see them, there are 2 centrioles that lie perpendicular to each other within the centrosome. The centrosome is a small clear area in the cell. You'll see it's a small clear area in the cell. Centrioles play a very important role in cellular division. And that concludes cellular structure. So we spoke about transport. We spoke about the different organelles of the cell. I'm going to place my email on somewhere where you can see it. And if you want the PowerPoint to this, please send me your email and I will send you a copy of the PowerPoint. Please like and subscribe, and thank you so much.